The modern dietary landscape, increasingly dominated by high carbohydrate and ultra-processed foods, is raising significant concerns about its long-term effects on brain health. While carbohydrates are a vital macronutrient and serve as the brain's primary energy source, the quality and quantity of carbohydrates consumed can make a profound difference in neurological outcomes. Diets heavily laden with refined carbohydrates, such as white bread, sugary beverages, pastries, and processed snacks, have been increasingly implicated in the promotion of brain inflammation, oxidative stress, and memory impairment. As evidence mounts, it is becoming clear that high carbohydrate eating patterns may not just affect waistlines and blood sugar levels, but could also accelerate the trajectory of cognitive decline. At the molecular level, one of the key concerns surrounding refined carbohydrates is their impact on blood glucose regulation. These foods cause rapid spikes in blood sugar, which, when sustained over time, contribute to chronic hyperglycemia and insulin resistance. In the brain, insulin plays a crucial role in modulating neurotransmitter activity, synaptic plasticity, and even the clearance of toxic proteins such as beta amyloid. When insulin signaling is impaired, as often happens in individuals with prolonged exposure to high glycemic diets, neurons can become energetically starved despite abundant circulating glucose. This paradox, known as brain insulin resistance, is now considered a possible driver of neurodegenerative diseases, particularly Alzheimer's disease, which some researchers have dubbed type 3 diabetes for its metabolic underpinnings. A further complication arises from the formation of advanced glycation end products, AGEs, which occur when sugars bind to proteins or lipids without enzymatic control. AGEs accumulate in both vascular and neural tissues and are particularly problematic in the brain, where they can cross-link with structural proteins, interfere with synaptic communication, and trigger receptor-mediated inflammatory pathways. The binding of AGEs to RAGE, the receptor for advanced glycation end products, on microglia and endothelial cells activates a cascade of pro-inflammatory cytokines, promoting neuroinflammation that undermines both cognitive function and brain repair mechanisms. This chronic low-grade inflammation may not produce immediate symptoms, but over time it erodes the structural integrity of key regions, such as the hippocampus and prefrontal cortex, areas essential for memory, decision-making, and emotional regulation. Oxidative stress is another downstream consequence of refined carbohydrate overload. Frequent postprandial glucose surges increase the generation of reactive oxygen species, ROS, which damage DNA, mitochondria, and cellular membranes. Since the brain consumes a disproportionate share of the body's oxygen and relies on highly sensitive lipid structures for signal transmission, it is particularly vulnerable to oxidative damage. In this environment, the capacity of antioxidant defense systems such as glutathione and superoxide dismutase can be quickly overwhelmed, leading to cumulative neural injury and hastening the aging of brain tissue. These mechanisms do not remain purely theoretical. Longitudinal studies have shown that individuals with diets high in sugar and refined carbohydrates perform worse on cognitive tests over time, even when adjusting for variables like age and education. Some neuroimaging data also suggest that excessive intake of these foods is correlated with reduced brain volume, especially in memory-related areas. Animal models mirror these findings. Rodents fed high-sugar or high-fructose diets exhibit impaired spatial memory, increased beta-amyloid plaque deposition, and signs of microglial activation consistent with neuroinflammation. But while the warnings are clear, it is equally important to recognize that not all carbohydrates carry these risks. Complex carbohydrates found in whole grains, legumes, fruits, and vegetables release glucose more slowly into the bloodstream, providing steady energy without provoking sharp glycemic spikes. These foods are also rich in fiber, antioxidants, vitamins, and polyphenol, compounds that help buffer against oxidative stress and inflammation. Diets like the Mediterranean or MIND, Mediterranean-Intervention for Neurodegenerative Delay diet, which emphasize plant-based complex carbohydrates and healthy fats, have been associated with slower rates of cognitive decline and reduced risk of developing dementia. There is also evidence that dietary patterns that reduce refined carbohydrate intake can reverse some of the inflammatory and metabolic markers associated with cognitive risk. Interventions such as low glycemic diets, intermittent fasting, and ketogenic regimens have been shown in preliminary studies to improve insulin sensitivity, enhance mitochondrial function, and reduce pro-inflammatory cytokine levels. Some of these approaches may even boost brain-derived neurotrophic factor, BDNF, a molecule that supports the growth and survival of neurons. Still, caution is necessary. Extreme carbohydrate restriction or unbalanced ketogenic diets may not be suitable for all individuals, especially older adults or those with underlying medical conditions. The brain does require a minimum level of glucose to function optimally, and any drastic dietary changes should be approached thoughtfully and ideally under medical supervision. Moreover, food quality remains as important as macronutrient ratios. Replacing refined carbs with processed meats or saturated fats does little to support cognitive longevity. In summary, the evidence increasingly supports a critical view of high-refined carbohydrate diets as contributors to chronic brain inflammation and memory loss. While glucose is essential to brain function, the form and delivery matter greatly. By prioritizing slow-digesting, nutrient-rich carbohydrates and minimizing ultra-processed foods, it may be possible to preserve cognitive function, reduce neuroinflammation, and support long-term brain health. This is not merely a nutritional concern. It is a matter of safeguarding one of our most vital organs against the slow, silent erosion of modern dietary excess.